Welcome to this presentation as part of Astrobiology Graduate Conference organized virtually by ELSI in Tokyo. I am Tomislav Stolar, a PhD candidate focused on prebiotic chemistry in solid state and working at Ruđer Bošković Institute in Croatia. First, I want to highlight the key message I want you to take away from this presentation. And this is that amino acids oligomerize to peptides on mineral surfaces, without the need to interact with water or aqueous environment. I want to acknowledge the amazing team of scientists with whom I worked on this project. Jose Gregorio Hernandez, a scientist with world-class talent and the originator of this project idea. Krunoslav Užarević, who is my PhD supervisor and my biggest supporter. The scientist with the right tools and solutions, who has an incredible passion for doing science. Nikola, Ernest and Sasha for offering us tremendous support and access to analytical techniques that we needed to carry out this project. Many thanks to Croatian Science Foundation for financing. To start with, the presence of amino acids on early Earth is consensually accepted by scientists in the field, either stemming from endogenous chemical routes or delivered extraterrestrially by meteorites. On the other hand, peptides are important biological polymers with many catalytic functions and are regarded to be included in ancient molecular symbiosis with nucleic acids. Here we have a molecular structure of the simplest amino acid called glycine. Generally, amino acids have an N-terminus with an amino group and a C-terminus with a carboxyl group. Peptides, on the other hand, are chains of amino acids linked together via peptide bonds, which is a covalent bond between carbon from a carbonyl group and a nitrogen from an NH group. How did nature go from amino acid to peptides is a major question in prebiotic chemistry, which is chemistry that led to life. The formation of peptide bonds is an example of a condensation reaction, where two amino acids, such as glycine, are joined together or condensed via peptide bond, together with the elimination of a water molecule. Looking at the basic Le Chatelier principle, if this reaction is carried out in aqueous media, it is highly thermodynamically unfavorable, because in order for this reaction to proceed, water molecules need to be eliminated. So, it seems that we have a water paradox. On one hand, water is essential for life and for biological evolution, but on the other hand, it seems that water is not desired for the synthesis of life's building blocks. This is not just related for the synthesis of peptides, but is a broader issue in prebiotic chemistry, because there are many condensation reactions, such as the formation of glycosidic or phosphodiester bonds. This has been recognized by more and more scientists in the field, and there is a growing realization that dry land was critical for concentrating organic building blocks and enabling their chemical evolution. There are different literature approaches for prebiotic peptide synthesis. Firstly, thermal polymerization, which requires high temperatures. Going back to late 50s, soon after the discovery of famous Miller-Urey experiment, which served as a birth of prebiotic chemistry. Secondly, hypervelocity impact studies that uses impacts in the range of kilometers per second and results in short peptides, such as di- and tripeptides. Thirdly, wet-dry cycling, which uses different approaches, but always uses water for mixing of the reactants and its subsequent evaporation in order for peptide bond to form during the dry phase. So we hypothesized, what if we avoid the use of water and instead turn to solid-state reaction environment and use only solid mineral surfaces? Our approach was to use mechanical energy that could have been provided prebiotically by impacts, erosion, earthquakes, and weathering, and to use thermal energy. The availability of dry land is often discussed in the field, and although its availability was only a few percentage of today's land mass, it is considered that there was some land available near volcanoes and on the emerging continents. We used mechanochemistry as a tool to carry out chemical reactions in the absence of solvents. Mechanochemistry has been previously used in prebiotic chemistry contexts for the synthesis of alpha-aminonitriles and sugars. We also used temperature-controlled ball milling setup developed recently by our group. We decided to use the simplest amino acid, glycine, due to its prebiotic availability, the presence in meteorites, and the availability of analytical techniques needed to characterize reaction products. Reaction mixture was closed in a milling jar and was ball milled via 3 stainless steel balls. The milling jar, together with the reaction mixture, was oscillated horizontally, and attached to it was a thermal ring element that was connected to a temperature controller. To characterize the reaction products, we used ion pairing high-performance liquid chromatography. As we can see on the chromatograms, raising the reaction temperature going from room 40 degrees, 70, 100 and 130 degrees Celsius, we have observed the formation of longer glycine oligomers. This is happening together with the formation of higher amounts of diketopiperazine, a cyclic glycine dimer that is sometimes regarded as a dead end for prebiotic peptide synthesis. The total yield of all linear oligomers was highest when the reaction was carried out at 100 degrees Celsius and accounted for 10%. In order to better understand the dynamics of mechanochemical peptide bond synthesis, we carried out reactions starting from dipeptide, tripeptide, and diketopiperazine. We can see on the chromatograms that for all these cases we observed the formation of odd and even number of oligomer length, meaning that the mechanochemical peptide bond formation is a dynamic process with simultaneous forming and breaking of peptide bonds. Importantly, when starting the reaction from diketopiperazine, we have ascertained that it can be a productive intermediate for the synthesis of peptides, as we observed the formation of peptides containing 10 glycine residues. In order to unambiguously determine the presence of different oligomer length, we have developed a method for ultra-performance liquid chromatography coupled with mass spectrometry. So first, to separate different oligomers and then to detect them using a Q-TOF detector according to their molecular mass. The longest oligomer detected by milling glycine with titanium dioxide was a peptide with 11 residues of glycine. This oligomer length is long enough for it to establish a three-dimensional structure, which is associated with the catalytic properties of peptides and proteins. To summarize our key results, we have successfully formed peptides when also using different mineral surfaces, such as silicon dioxide, montmorillonite, and mica. Mechanochemical peptide bond formation tolerates the presence of moisture, as verified by performing liquid-assisted grinding experiments where we added small amounts of water. This was to show the robustness of mechanochemical route that does not require glove box conditions. that were certainly not available on an earlier scenario. We also tried to use different amino acids, such as L-alanine, and after milling it with titanium dioxide, we have detected its pentapeptides. When milling together glycine and L-alanine with titanium dioxide, it resulted in the formation of their heteropeptides, and we have successfully shown that it is possible to incorporate other amino acids into a growing peptide chain. Again, I want to highlight a key message for you to take away from this presentation, and this is that amino acids oligomerize to peptides on mineral surfaces without the need to interact with water or aqueous environment, using only the input of mechanical and thermal energy. Finally, what are the implications of mechanochemical route towards peptides for astrobiology? Can we expect to find peptides in asteroid samples? These are some of the very exciting questions. Please do get in contact with us if you have any additional questions or suggestions. And many thanks for your attention.